Hello friends, welcome to my YouTube channel. My name is Mohammad Sohil and in this video we are going to learn human health and disease. In this video we are going to see what do you mean by immunity, what are the different types of immunity and what is antibody. So without any further delay, let's start our today's session. So first of all, what do you mean by immunity? The ability of an individual to resist foreign agents or microbes which cause disease is called as immunity. Now what is immune system? The system which protects us from various infectious diseases is called as immune system. Then third, immunology. The branch of biology which deals with the study of immunity system and immune response is called as immunology. Now coming to the concept of immunity. So in Latin, immunis means save from something. There are branches related to immunology. First, clinical immunology. It includes vaccination, organ transplantation, blood banking, etc. Then laboratory immunology. It includes testing of cellular and humoral immune functions. The most important characteristics of immune system is to identify self, that is own body cells, and non-self. It also keeps immunological memory. Human immune system mainly depends on antibodies which are produced against antigen. Now what is antigen and antibody? So antigen, any foreign material that stimulates the production of an antibody in an animal is called as antigen. Then second, antibody. A specialized glycoprotein produced in response to a particular antigen is called as antibody. Now coming to the different types of immunity. So immunity is broadly classified into two types. Innate immunity, which is also called as non-specific or native immunity. Then second, specific immunity, which is also called as specific or adaptive. Now coming to first type, that is non-specific immunity. All organisms have naturally gifted with resistance to certain infections. This immunity is present since birth. This is called as innate immunity or inborn immunity. This immunity is present due to genetic constitution of organism. It is not dependent on the prior contact with microorganism. It is the first line of defense of most of the organism. Innate immunity consists of following barriers. Now coming to the barriers of innate immunity. So there are four types of barriers in innate immunity. First, anatomical barriers. Second, physiological barrier. Third, phagocytic barrier. And fourth is inflammatory barriers. Let's see one by one. First, anatomical barriers. These barriers prevent the entry of microorganisms into the body. These barriers are skin and mucous membrane. Mucus trap microorganism. Example, epithelial lining which produce mucus is present in gastrointestinal tract. Now second barrier, that is physiological barriers. Physiological barriers like body temperature, pH, body secretion, etc. It prevents the growth of many microorganisms. Body show fever when pathogen attack on body. Fever is high temperature which inhibit the growth of many pathogens. A strong acidity of gastric juice kills most of the ingested microorganisms. Lysosome is a bacteriolytic enzyme which present in tears. Then third barrier, that is phagocytic barrier. Phagocytes are specialized cells of our body which engulf and destroy the microorganism. Thus they protect our body during infection. The number of leukocytes increase greatly. The most important phagocytes are macrophage, neutrophils and monocytes. Then last barrier is inflammatory barriers. Inflammation is non-specific defensive response of the body. Inflammation is caused due to tissue injury, insect bites, certain chemicals or by virus, fungi, bacteria, etc. The inflammatory area shows some characters like swelling, heat, redness, pain, etc. This may cause fever. Such localized inflammations are called inflammatory response which prevent from pathogens and toxic chemicals. Now coming to the second type of immunity, that is acquired immunity, which is also called as adaptive or specific immunity. The immunity which is required by an individual during his lifetime is called as acquired immunity. It also known as specific immunity because specific antibodies or lymphocytes are produced for a specific antigen. It is not present since birth. Now coming to the types of acquired immunity. There are two types of acquired immunity, that is active acquired immunity and passive acquired immunity. Let's see first. That is active acquired immunity. Active immunity is generated when antibodies are produced by B lymphocyte in the body's own tissue when they encounter antigens. There are two types of active acquired immunity, natural active immunity and artificial active immunity. Now what is natural acquired immunity? This type of immunity comes when infection is done by bacteria or virus. During infection, antibodies are produced against this pathogen. This is first response to antigens, which has never infected to the body earlier. If the infection of same pathogen is occur again, the disease will not affect the person because the antibodies are already present in the bodies. Now second type of active acquired immunity is artificial acquired active immunity. This type of immunity is introduced in the body through vaccination or immunization. A small amount of antigen, that is dead or extremely weak, are introduced into the body as vaccine. This induced response and antibodies formation takes place against antigen. As a result, the body has sufficient antibodies to control the disease in same pathogen infect again. Now coming to the second type of acquired immunity, that is passive acquired immunity. Passive immunity is developed by the organism as a result of transfer of antibodies which are developed in another organism. It is of two types, that is natural passive immunity and artificial passive immunity. First, natural passive immunity. In this type of immunity, the antibodies are transferred by natural way. Antibodies are naturally transferred from mother to developing fetus. After birth, they are transferred through colostrums. The antibody IgA is received from mother by child. This antibody remains for short time, thus natural acquired active immunity remains for the short time. Then second, artificial passive immunity. This immunity developed due to transfer of antibodies in the body which are developed in another individual. This transfer is done by giving injections. Example, antibodies for tetanus is developed in horse, then they are transferred into human body. Now coming to the difference between innate immunity and acquired immunity. So innate immunity is present from birth, which is contact with pathogen is not necessary. It is inherited from parents. It is not related with the previous attack of disease. It consists of various barriers which prevent the entry of pathogens in body. Then acquired immunity. This is developed after birth. Contact with pathogen is necessary. It is not heritable. It is related with the attack of disease or vaccine. It consists of specialized cells like beta lymphocyte and T lymphocytes. Coming to the last topic of our today's session is antibody. So antibody is a specialized glycoprotein produced in response to a particular antigen is called as antibody. Now coming to the structure of antibody. Antibodies are glycoproteins. They are highly specific to antigen. They are also called as immunoglobulins, that is IgS. They are produced by plasma cells. Per second about 20,000 antibodies are produced. It is a globular Y-shaped proteins. It consists of 4 polypeptide chains, 2 identical light chains and 2 identical heavy chains. Light chain has 2 regions, variable regions V and constant region C. A light chain is composed of about 200 amino acids. The variable region consists of about 1 to 108 amino acid. It forms antigen binding site. The constant region consists of 109 to 200 amino acid. The sequence of amino acid in constant region is identical in different antibodies when it belongs to same class. Now coming to heavy chains. Heavy chains is composed of about 440 amino acids. It has both variable region and constant region. The variable region consists of about 12118 amino acids. The constant region consists of 119 to 440 amino acids. Both two light chain and two heavy chains are linked together by 3 phosphodiester bond to form Y-shaped molecule. The tip of the variable region is both light and heavy chains form two antigen binding site, that is paratops. It forms an antigen antibody complex. So in this way we complete our three topics, that is immunity, different types of immunity, and antibodies. So please share this video with your friends and subscribe to my channel for more informative videos.